Hi, my name is Ecaterina Dumbrava. I'm an assistant professor in the Investigational Cancer Therapeutics Department at MD Anderson Cancer Center in Houston, Texas. And I will be presenting on the topic of HER2 targeted immunotherapy. Here are my disclosures. Um, as a quick background, HER2 or the human epidermal growth factor protein 2 overexpression is a proven predictive and prognostic biomarker that was among the first actionable targets for targeted therapies. Without doubt, the current oncology treatment has been focusing on personalizing cancer treatment based on the tumor genomics, and treatments are becoming more tumor agnostic. And so, treatment with the immune checkpoint inhibitors changed the way how we treat several tumor types. And these treatments showed long and durable responses with manageable adverse events. However, outside some markers such as MSI high tumor mutation burden, and in some cases PD-L1 expression, there are no great targets or ways to select patients for immunotherapy or personalized cancer immunotherapy. So, taking an established cancer biomarker such as HER2 overexpression that is very specific for tumors and developing treatments that engage the immune system will, with the promise of better and more durable responses, seems a logic thing to do. And so HER2 alterations are present not only in breast and gastric cancer, and we show that HER2 amplifications by next generation sequencing is present in about 5% of patients with biliary cancers. Here you can see on the screen that we have approximately 5% of HER2 amplifications in biliary cancers, including both gallbladder and cholangiocarcinoma. And we showed that HER2 targeted therapies can improve survival and outcomes of patients who are HER2 positive, regardless of the tumor type and beyond breast and gastric cancer. And there are many HER2 targeted therapies, including antibodies, small molecules, antibody drug conjugates that you can see on the screen. And most recently, there has been an increasing interest in targeting HER2 and immune cells, and that will be the focus of our talk today. And so, first, I want to discuss briefly about the margetuximab, which is the newest kid on the block of HER2 targeted therapies that was approved in December 2020. And it is an FC engineered HER2 directed monoclonal antibody, and that binds to the extracellular domain of HER2. And upon binding to HER2 expressing tumor cells, margetuximab inhibits tumor cell proliferation, reduces shedding of HER2 extracellular domain, mediates antibody-dependent cellular cytotoxicity, and in vitro, the modified FC region of margetuximab increased binding to activating FC receptor of CD16A and decreased binding to inhibitory FC receptor CD32B. These changes led to greater in vitro antibody dependent cellular cytotoxicity and also to NK cell activation. However, the clinical significance of it's still unknown. The efficacy of this drug was evaluated in the SOPHIA trial, which is a randomized multicenter phase 3 clinical trial evaluating 536 patients with HER2 3 plus by immunohistochemistry or FISH amplified and 2 plus by immunohistochemistry who have received prior HER2 targeted therapies. And in the study, the randomization was 1-to-1, and 266 patients were treated with margetuximab plus chemotherapy, and 270 patients received trastuzumab and chemotherapy. The goal of the study was to demonstrate the efficacy of the experimental margetuximab combination, which could offer a new treatment option for these patients who are refractory to prior HER2 targeted therapies. And so, to demonstrate the efficacy, the study explored as the primary endpoint the progression free survival and the overall survival. The final analysis, it's for the overall survival, it's still pending. However, the treatment was approved based on the results for the PFS, showing that the combination of margetuximab plus chemotherapy, capecitabine, eribulin, gemcitabine, or vinorelbine, showed an increase in progression-free survival from 5.8 months compared to 4.9 when compared with the trastuzumab plus chemotherapy. And for the final results of the overall survival, they are still pending. Second, there is a new class of treatment that offer to targeted immunotherapy that is called immune stimulator antibody conjugates, or ISACs, which are systemically delivering an immune modulator such as a TLR7/8, and that is conjugated to a HER2 targeted antibody. And I listed here the three more that are currently in development. And so BDC-1001 is a novel immune stimulating antibody conjugate, or ISAC, that consists of an investigational biosimilar of the humanized monoclonal antibody trastuzumab that is conjugated to a TLR7/8 agonist with a non-cleavable linker that is administered systematically IV every 3 weeks. And ISACs such as BDC-1001 initiate an entirely new response by activating the human myeloid antigen presenting cells while retaining the antibody-mediated effector functions such as antibody-dependent cellular cytotoxicity and phagocytosis. And studies in trastuzumab-resistant xenograft models and syngeneic tumor models indicate that HER2 targeted ISACs can elicit a potent and durable response, leading to complete tumor regression in preclinical models. And so for the phase 1 study with BDC-1001 is ongoing, and it's enrolling patients with HER2 expressing or HER2 amplified advanced or metastatic solid tumors. And for those expansions, we'll have specific cohorts, including HER2 low breast cancer. And another ISAC, the NGH395, is also a HER2 targeted antibody conjugated with a TLR7 agonist via non-cleavable linker, for which the preliminary results were presented last year at SITC, and for 18 patients were treated. And as opposed to BDC-1001, which is given IV every 3 weeks, patients receive a single dose, and the patients were followed for tumor progression at 3 weeks after the infusion. And more than a half of patients presented a cytokine release syndrome that was a grade 1 or 2, and other mild adverse events. Nine patients presented stable disease per RECIST at 3 weeks post-treatment, and no complete or partial response were seen. However, what is encouraging is that CD8 positive T cells infiltration of the tumor was seen in the tumor biopsies on treatment in five of the patients. Another ISAC, the SBT 6050, is also a HER2-directed monoclonal antibody conjugated, this time with a cleavable linker to a potent specific TLR8 agonist. And it's also in a phase 1 clinical trial in combination as monotherapy or in combination with pembrolizumab. And so uh, I will tell you more about HER2 CAR T cells. And so these are there has been a lot of excitement 10 years ago for developing HER2 targeted CAR Ts. However, after a fatal case of a 39-year-old patient with HER2 positive colorectal cancer with a third generation HER2 CAR, after that, there has been a lot of pushback, and all trials were conducted very carefully with HER2 CAR Ts. And Ahmed and all conducted a second generation CAR T targeting HER2 for patients with sarcoma without leukapheresis or lymphodepletion, and 19 patients received the treatment with two partial responses and four stable diseases. And in another study, Ahmed and all, the same team from Baylor, also tested the HER2 CAR T in patients with glioblastoma without lymphodepletion, and this was conducted without any serious adverse events. And in both of these studies, there was a persistence of the engineered T cells up to 12 months, but no expansion, which highlights the importance of the lymphodepletion. The lymphodepletion prior to the T cell administration improves the expansion of adoptively transferred T cells, in part due to increased availability of the homeostatic cytokines such as IL-7 and IL-15. And in addition, the lymphodepleting chemotherapy decreases both the Tregs and the myeloid-derived suppressor cells, thereby augmenting the expansion of T cells and increasing the anti-tumor activity. And some encouraging results were seen of a 7-year-old boy with metastatic rhabdomyosarcoma that was reported recently after confirming the HER2 expression on both on the primary tumor and the bone marrow metastasis by immunohistochemistry. The patient was enrolled in phase 1 clinical trial with HER2 CAR T, and he received the HER2 CAR T that was manufactured, and the patient received the treatment 4 weeks after washout from prior chemotherapy, and he received 3 infusions of the HER2 CAR T cells 10 weeks apart, each after lymphodepletion with cyclophosphamide and fludarabine. And at the end of this induction phase, no morphologic or imaging evidence of rhabdomyosarcoma were seen. And so thereafter, the same dose of CAR T was administered for 6 additional months with the intent of consolidating the response to this treatment. And so, what is important for this audience is also this this clinical trial that had a modified second generation CAR with with a proprietary and ectodomain in 11 patients who had lymphodepletion with nab-paclitaxel and cyclophosphamide. And patients received 1 2 cycles of cell infusions, and most side effects were mild, such as fatigue and lymphopenia. 1 patient had a partial response, and 5 patients had stable disease, and 3 patients had a progression free survival of more than 5 months. And 5 of the 11 patients showed evidence of in vivo expansion of T cells. And so that leads us to new type of cars and designing better HER2 cars in which there is a control by molecular switches which are introduced to the T cells along with the CAR during the gene modification. And these switches can regulate both the safety and the activity of CAR T cells post infusion and has the potential to improve the therapeutic profile. And so this is BPX603, which is an autologous T cell product that is genetically modified by retroviral vectors containing a rimiducid inducible coactivation molecule, and also a first generation CAR and the Rapalog inducible caspase 6, which is the safety switch, and the truncated CD19 that is used as a selection marker. And the Go switch that activates the T cells and their proliferation and survival is using rimiducid, which is a small molecule that helps the dimerization of the IMC and induces the downstream signaling cascade and improves the BPX603 proliferation and persistence. And for the safety switch, it is used temsirolimus, which can activate the IRC9 and can induce apoptosis and reduce the cytokine and mitigate acute toxicities. And there are many other HER2 immunotherapies. Unfortunately, I will not have the time to go through all of these. However, I will just mention some that they are promising and they are currently in clinical trials, such as bispecific antibodies, such as HER2 and 4-1BB, HER2 and CD3, also HER2 targeted NK cell therapy, and some vaccines that they are in development. And so, as take home messages, HER2 is a proven biomarker, and also there have been several HER2 targeted therapies approved for breast cancer and for gastric cancer. For these patients, unfortunately, they still progress. And we need better treatment options. And targeting both HER2 and the immune system seems a promising way to induce durable responses. However, we have many things to learn and understand the immune profile of patients who are HER2 positive, and we need better translational studies in which we look at mechanism of resistance and response to HER2 targeted immunotherapies. And also we need to design better clinical trials, and that would lead to better treatment options approved for patients. And hopefully we will be able to have personalized immunotherapy treatment options for our patients in the future. Um, and here is my contact information. The phone number is my cell phone, so feel free to reach out to me if you have any questions.